Alright, so we're going to continue on our discussion with genetic cloning. For a human gene to be expressed in bacteria, it's going to have bacterial regulatory regions, meaning it's going to have bacteria promoter regions, bacteria stop codons, start codons, all of those things to help regulate the expression of that gene. Also, interestingly enough, bacteria do not have introns, so the gene itself could not contain introns, or the bacterial polymerase will not actually synthesize the DNA. Will not synthesize the new protein. Enzymes called reverse transcriptase are going to be used to make the human gene that lacks introns. What they're going to do is take copies of the mRNA into complementary DNA, which would not contain the introns. So you sort of create. You take the RNA and then you create a section of DNA based off of that. The recombinant cells make many copies themselves, each containing the new gene. So we only need to make one copy of that gene and then introduce it to that bacteria. Each new cell is going to make the product coded for by that gene. So if we code for a section of DNA that's going to produce insulin, the bacteria themselves will make multiple copies of that gene and then also start to make that gene product. They will make that insulin themselves. So very important part of cloning would be DNA editing, the ability to edit and change certain portions of the DNA. So genome editing is just the targeting of specific sequences in DNA for removal or replacement. A lot of times, the methodology that's going to be used is CRISPR. This is clustered, regularly interspaced short palindromic repeats. This uses a specific enzyme, the Cas9 enzyme, which is going to identify specific nucleotides that are going to be cut out using a guide RNA molecule. So here we have the original gene. We have our DNA that's going to be cut out and removed. We have our Cas9 enzyme that is going to identify the specific gene sequence that we want. It's going to enable us to cut out or remove the section of DNA that we need, which can then be inserted into the new DNA. In order to protect the bacteria from our Cas9 enzyme, we're going to need a sequence called PAM. That PAM sequence is going to be placed next to the larger DNA sequence that we're going to use. The insertion of that sequence is not found in bacterial cells, so the bacterial, this is the PAM sequence. Without that PAM sequence, the Cas9 is not going to start cutting DNA. So Cas9 is not going to connect to the bacterial DNA because they do not have this initiating sequence, that PAM sequence. The reason we care about this is because we want to create different biotechnology products. Biotechnology products are just produced by cells that have been genetically engineered. DNA is going to be altered so cells can produce a specific substance. Transgenic organisms are simply organisms that have had foreign genes inserted into them. So, for example, the bacterial cells that we have been talking about are using as an example. When we insert human DNA into them, we have created a transgenic organism. Recombinant DNA technology is the process by which we produce those transgenic bacteria. So naturally occurring bacteria that eat oil can be genetically engineered to eat even more oil to help clean up beaches after oil spills. This is a really common use of transgenic organisms. There are bacteria out there that naturally will eat oil and clean up after oil spills. However, they won't do enough to make it super efficient, so we can genetically modify them to absorb even more oil. Then those bacteria are given suicide genes so they can self-destruct when they are done. In other words, they're given the directive to apoptose, so that way they will break down in safe ways and permanently remove that oil from the area. Most corn, potato, and soybean and cotton plants have been engineered to be resilient to either insects or herbicides or both. Genetic engineering can also decrease food spoilage. If you knock out a gene that causes browning in apples, it will increase its shelf life. So here's an example of apples that have had the gene that removes to oxidize when exposed to air. So the apples simply don't brown as quickly, and it helps them to survive longer on the shelf. Soybeans can be engineered to produce a beneficial fatty acid, which can help improve health. Leaves can be engineered to lose less water and take in more carbon dioxide. This could help crops grow in a variety of climates where water is maybe not as plentiful. Other modifications allow plants to produce hormones, clotting factors, antibodies, vaccines. Animals can be genetically modified to increase their value as food. Transgenic salmon produce a growth hormone that could cause even faster growth. They could also be engineered to be sterile. A transgenic form of mosquito that carries the Zitka produces proteins that will kill off their own offspring. So here's an example of a transgenic mosquito. So they're going to, so it'll kill off their offspring so it can hopefully stop the spread of the Zika virus. Gene farming is the use of transgenic farm animals to help produce pharmaceuticals. Genes that code for proteins are incorporated into the animal's DNA, and then they can appear in the milk of the animal. So cloning can then be used to produce many animals that will, or cloning can be used to produce many animals that produce the same product. The milk could then contain antibiotics, vaccines, or they might just even have a higher nutritional value. Mouse models have been used to study human diseases. We can clone certain genetic disorders into mice and then study the way those diseases progress in the mice. The cystic fibrosis allele has been inserted into mice. The mice then develop cystic fibrosis. The mice that carries the genes, so then we can study various cystic fibrosis treatments on that particular mouse model. Xenotransplantation is the transplantation of animal organs instead of human organs. So it's another way to create transgenic organisms is simply the transfer of organs. This will occur. We'll see this frequently in heart valve replacement. Xenografts can be used to replace or graft animal organs, like animal heart valves can be used to replace human heart valves if that is what's determined to be best for the individual. So coming out of this section, I really want you to be familiar with how the polymerase chain reaction works and how recombinant DNA works. You should have a general idea of how genome editing can change a nucleotide sequence. You should also summarize the reasons that GMOs are produced. But the biggest thing I want you to focus on this section is the process of recombinant DNA. I want you to know the terms vector, plasmid, recombinant DNA, host cell. I want you to know the role of the restriction enzymes. So go through that whole process of recombinant DNA formation. Let me know if you have any questions. I'll be happy to go over anything with you in greater detail.